So far, we were talking about the mRNA synthesis mainly. I use the word transcription, but of course, it's unavoidable. But here, we will use the real word in real sense as a transcription, because we are going to talk about the DNA to RNA, which is the norm in the eukaryotic cells. And the part 2, we will try to do the part 1 today and do the part 2 in the next class. So that's about it. So you will just have only one quiz due on the topic 6, not this one. So in the first part, what we are looking at, that if you just look at the yellow ones, these viruses do have DNA in their life cycle. Even the retrovirus is a plus RNA virus, but by the reverse transcriptase, it makes a minus DNA intermediate, then make a plus DNA. So from the plus DNA, it's going to make the mRNA the same way. If it is a minus strand DNA for these, or the plus strand DNA, they are going to be converted into double-stranded. If it's a gapped, the same way. And of course, if it is a double-stranded DNA like the adenoherpes or the polyomo virus, they are going to transcribe into the mRNAs no matter what. So all of them are going to make the mRNA, and we will see that for each one of them, how they reach that goal. That's the basic idea of the whole thing. So the first thing that what we are looking at, that in the cells infected with the DNA virus, at least one protein is needed for the DNA replication, and many, sometimes often. So, but at least one, this is very important, at least one protein. Even if that virus is using the host machinery, but it needs one. And without the mRNA transcription, viral protein cannot be synthesized. And this is the rule. There is no exception. There is at least one protein need to be made before it can be done. But what happens, that the DNA templates are not ready for transcription when it is delivered into the cell. The reason that in the cell, you need a certain way, the genome needs to be packed in a certain way, so that the RNA polymerases can actually transcribe them. So that's very important. But some of them are ready. Say for an example, right here we are looking at the example of this SV40, this polyomavirus, and you can see that this is the structure of its genomic DNA. And this is transcription ready. It readily is transcribed into the mRNAs. So you have a unique structure with which it comes in. And what is that structure? Anyone? That the roundish structure looks like your, right here, looks like a donut. Anyone? That's basically your histone octamers. That's your nucleosome. So if the DNA is packaged into the nucleosome, you know that you have the H2A, B, and then you have the H3 and H4, the 4 different types of histones, and they are the octamers of those. And you have about 149 base pairs wrapped around them, of the DNA wrapped around them. And if the DNA is in the nucleosomal format like this, this is like the eukaryotic DNA. If the cell receives a DNA from the virus like this, it can go into the direct transcription. But this is not the case in most cases. So that's what we're going to see. So how this happens. For the hepatinovirus, you can see that there is a discontinuous, it's a gap DNA. One of them is partial with an RNA. So a cellular enzyme actually makes the DNA completely double-stranded. And this mechanism, how this cellular protein does this process, is not completely known. It is still a new virus, and it's still being worked on. Parvovirus have a single-stranded DNA with a 3' open OH, and the whole thing is circularized by this. And after this recircularization, it also is made into the double-stranded. So always that is also repaired. In the retroviruses like HIV, reverse transcription makes the first copy of the DNA, then it makes the same enzyme, makes the second copy of the DNA, and it is usually integrated into the genome of the host, from where the cellular transcription machinery will make the mRNAs. But no matter what, the first thing that is needed for all the viruses which are not ready, unlike this SV40, they needed to be repaired or put into a system to be read by the RNA polymerases. And these RNA polymerases are mostly of the host origin, except from some of the large viruses can make the RNA polymerases. But these are, when they are read by the host RNA polymerase, they needed to be converted. Now, one of the most important thing that I just wrote it down very clearly, that remember that whenever you see the word RNA polymerase and it is not broken down, you check a couple of things to understand that what exactly it is doing. So if it is an RNA polymerase and if you see that it is an RNA virus RNA polymerase, remember that it will be always, without any exception, an RNA-dependent RNA polymerase that we are talking about. Again, repeating, if it's an RNA virus and we are referring an RNA polymerase, without exception, it will be an RNA-dependent RNA polymerase, always of a viral origin. But if we are referring an RNA polymerase and you see the virus is a DNA virus, most of the time we are referring to a DNA-dependent, remember that, DNA-dependent RNA polymerase, or DRP, capital D, small d, RP. Instead of RDRP, RNA-dependent RNA polymerase, for the DNA virus, you have a DNA-dependent RNA polymerase, which is the RNA polymerase of the eukaryotic host cell system. Basically, there are several one of them, and we're going to see this just only for few of the giant viruses, or say pox viruses, just a few of them. The DNA-dependent RNA polymerase is of the viral origin, but most of them are of the host cell origin. So that's one of the important conception, and I have written in the very first paragraph itself so that you remember that. So what are the eukaryotic DNA-dependent RNA polymerases? And they all can synthesize de novo, no primer needed. RNA pol, we are not going to talk about that, not known function. RNA pol 2 is the most important one. And see that what it does, it makes the pre-mRNA. Pre-mRNA means mRNA. Remember that all the pre-mRNAs are made with the introns and exons in eukaryotic cell. They are spliced out, or the exons are spliced out, introns are joined. That's become the mature mRNA. So pre-mRNA is basically the making of the mRNA in the nucleus. On the other hand, the RNA, these are cellular functions, remember that. Then the primary microRNA is usually about 70 to 100 base pair long. And these are processed, and finally the final product is 18-20 base pair long, or 18 to 25 base pair long. And the microRNAs basically have complementary sequence to gene sequences, and these work to silence gene expression. So these microRNAs are going to bind to the specific sequence on the DNA. This is like your siRNA, silencing RNA, but this mechanism is different. The siRNA has a bacterial origin. Here it is different, and that the microRNAs also silence gene expression by this process. RNA polymerase II make that one as well. And also it makes the small nuclear RNAs. These are sometimes also referred to as spliceosome-related enzymes, and they have a ribozyme function. They have catalytic RNAs sometimes. So these are the cellular function of the RNA pol 2. And for the virus, what they make, they also make the primary mRNA, which is spliced. They also make the primary miRNA, and these are also have a silencing function. And also, this is definitely underlined that this is very interesting. The hepatitis delta virus has an RNA genome. This is the only exception where a DNA-dependent RNA polymerase can read that RNA genome of the HDV genome to make its genome RNA as well as an RNA. So think about that. I'm repeating again. Only in the case of HDV, hepatitis delta virus, I've written that whole thing down in there. The DNA-dependent RNA polymerase, or the RNA polymerase II, functions as an RDRP, RNA-dependent RNA polymerase. There is no other known examples of that except the hepatitis delta virus. So that's very, very unique and always a quiz or exam question. that so definitely remember that. Then you have the RNA polymerase 3. These makes the tRNAs, 5S ribosomal RNAs, and also makes the U6 snRNAs, small nuclear RNAs. These are also found in the spliceosomes. So these make some very interesting RNA. These also make the adenovirus associated, virally associated RNAs. They also make the Epstein-Barr virus encoded RNA, encoded region RNAs. MHV68 is a murine virus, herpes virus. They also are responsible for these primary microRNAs to silence some of the eukaryotic gene expression. So they are not, so RNA Pol 3 doesn't make any mRNAs. Very carefully see that. RNA Pol 2 make mRNAs for most of the RNA viruses, DNA viruses. Most, except the ones that doesn't have, have their own. The RNA Pol 3 does not make any mRNA, but these are additional RNAs. They are not transcripts, but they do several different things. Many of them do have pathogenic effects. They do have anti-immunogenic effect, and we are going to talk about that in somewhere else as well. And one of them is very clear. You can see that this microRNA is a silencing RNA, is going to suppress gene expression of the host. So these are some of the functions for these RNAs. All right, so here also we talked about that only the DNA virus that replicate in the cytoplasm is the pox virus and the giant viruses. These are your Mimi viruses, Pandora viruses, Pithoviruses that we talked in the first class. These are the only viruses where the DNA-dependent RNA polymerase is made by the virus, and that's the reason they do not need to go into the nucleus to replicate. Other than that, all DNA viruses need to be delivered, or their genome need to be delivered into the nucleus for their replication, because that's the location for your RNA polymerase 2, 3, or all the DDRPs, DNA-dependent RNA polymerase. No DNA virus replicates in the cytoplasm except that last line. Pox are the giant viruses, and the reason you know that, they code their DNA-dependent RNA polymerases. Don't use the host polymerase. All right, so here, this is just overview. We are just only going to talk about the transcription in the first part. The rest, which is capping and splicing, export, we are going to, these are the post-transcriptional modifications. We're going to talk as a second part of this. So that's basically what it is. So the first thing is that the transcription is extremely or highly regulated. The thing is that this will give you an idea that how it is. You probably have 25,000 genes in every cell. But out of those 25,000 genes, at one point of time, you have about 5,000 to 8,000 of them are functional. And that also varies. When you're young, there are some of the genes that are active. When you're adult, something happens. If you have a stimuli going in after a meal, some of the genes are expressed, some of them are turned down. I mean, like, it's an extremely complicated transcriptional regulation in the eukaryotic system as well. So to do that, there is an extensive replication, and believe me that although it will look a little bit intimidating in the next few chapters and looks like that's a lot, literally it's the tip of the iceberg of the total transcriptional control that is known to us. Many things we probably don't know, but there's a ton of information on these areas. So this highly regulated system, they do have a general scheme, and this is what we are looking at. And what we are looking at, the yellow region, first, if you just look at the yellow region, the +1 with this red sign, this is a very common thing, that +1 is the start site of a gene. Remember that. That's the first A. All code mRNAs will start with all the proteins will start with an AUG. But remember that the mRNAs may not start at the AUG. Before the AUG, there could be some sequences. So remember that. But it is the first base of the mRNA. So that's the important part. Because the reason that I'm just not want to go off too much, because before the first codon, you need a specific sequence. Anybody knows what is that the ribosome recognizes other than the cap? from your micro? Shine-Dalgarno sequence, anyone? So you have a Shine-Dalgarno sequence which is recognized by the ribosome that is upstream of the first AUG, and those are in those places. So this is your +1 start. This is an initiator sequence, and there are some, so this is your DNA, and some DNA binding proteins bind in there. Then the next most important sequence is the TATA box or the TATA sequence. And why is it named? Because you have repeated TA residues in there. And in the TATA box, some of these transcription factors will bind. Remember that when I'm referring transcription factors, these are all DNA binding proteins. These are all DNA binding proteins. So they bind to the initiator, they bind to the TATA, and we're going to see that other regions as well. The TATA sequence, if you have taken microbiology in bacteria, you have a similar sequence. It's not called a TATA box or TATA sequence. It's called something else. Anyone remember? 3050? No? It's called the Pribnow box. Anyone remember? It's called the Pribnow box instead of TATA sequence in bacteria. So this TATA sequence binds to a transcription factor which is known as the TF2D. There are literally like hundreds of transcription factors. I will just let you remember just only two, the TF2D and the TF2H. It's in the next slide. But the TF2D binds to the TATA sequence. And then what you are looking at, that the local regulatory sequence, there could be some, and these are all will bind to DNA binding protein. And then you have this distant regulatory sequence which could be even 10,000 base pair away from the start sequence of the gene. And they do not require any specific orientation. They usually loop out and interact with the initiation sequence. So what are the sequences? So the TATA sequence and the initiator sequence make the core promoter. RNA polymerase binds onto this. But if there is nothing happens, RNA polymerase is usually extremely lousy to go on or move on. It's not going to work. You need the total regulatory sequence to at least start the RNA polymerase to do the transcription. If you have the enhancers, that will even augment the transcription, or these could be silencers as well. These elements, when bound, they can actually stop the RNA polymerase to transcribe. So the effects could be both ways. So that's your transcriptional control region. Another thing is that the distant regulatory sequence, it shows here. So if this is the start of the gene, this looks like it is downstream, but remember that it doesn't have to be that way. It could be anywhere. It could be in this direction as well, 10,000 base pair away, 5,000 base pair away, somewhere else. It doesn't matter whether it's upstream this way or that way. That's it is. So these are some of the basic sequence. So know the core promoter contains what? TATA sequence, initiator sequence, bind to RNA polymerase. Local regulatory sequence are within these 100 to 1,000 base pair, and usually the distant regulatory sequences are much farther position and orientation independent. All right, so here on the other hand, you are looking at some of the regulatory sequence of transcriptional control region. And if you just ask that what I need to know from here, literally nothing. Just giving you an example in there that this is an 82 adenovirus major late promoter. This is a gene, and this is their sequence. So it's different. It is not that every one of them are similar. For SV40, this is the gene start site, multiple start sites, just not only one. And you can see that this is the TATA box right here. The olive is a TATA box. It is the same everywhere. And this is another site which is called this SP. It's not SPL. It is the SP1. Remember that. These are the repeated sequences one after another that binds to different transcription factors. Whereas see that this is the adenovirus major late promoter. An early promoter does have a different sequence. And here we are going to see that this is the E2F binding site. E2F, we are going to see later part, is a eukaryotic transcription regulator. And we are going to see the function of this E2F. So that's important. So just remember that they are extremely diverse, although they have a specific type. So you have an initiator sequence. Here you do not have an initiator sequence, but local regulatory sequence, but all of them have a TATA sequence. That's your take-home message from this slide. That should be good enough. So here we are looking at the function of a distance activator, and there are two types of these. One is called a cis-acting element if they are close to each other. So this is your regulatory sequence, and if the cis-acting elements are close, these two proteins bind and interact together. They're on the same side. On the other hand, a trans-acting element is where you have the protein much binding to the DNA, much longer distance. This is what we call as tens of thousands of base pairs away. And a DNA loops out to interact with the element. And this is what we call a trans element. So definitely underline that. You will see some pictures in your exam, and you have to figure out whether it's a trans-acting or the cis-acting element. So these pictures are very important for that. So this is how in reality it looks like. And again, as I said, that there are only two things that is needed. So if you just look at the RNA polymerase II, it has the 12 to 16 subunits of that. And many of them are conserved. Just a few subunits here and there makes an RNA pol 2, 1, and 3. So that's very interesting. And you do not have to remember any of these. So just remember that your TF2D that binds to the TATA box, and the TF2H. That these are the two regions of this that you know that. And then you can just easily see that your enhancer binding proteins, how they are binding to base pairs very distantly, and they can act on the RNA polymerase. Just remember that all these proteins, subunit that you are seeing, not even all of them are shown. As I said, they're usually 12 to 16. Just a couple of them are shown. This is basically the RNA polymerase, and these are the transcription factors. And that's basically the overview of the whole thing. All right, so now the proteins that regulate this transcription, and we call these as a transcription factors, transcriptional regulators, transcriptional elements. So any of these words that you figure that if it is a transcriptional element, usually most of the time it is talking about the DNA. So first, when you encounter the term, just try to understand that what is being meant, whether it is meant by the DNA, transcriptional region, is it a DNA? If it is a DNA, DNA. On the other hand, if you see the term transcriptional factor or transcriptional regulator, usually it will be a protein which is a DNA binding protein binding to those transcriptionally active site that we talked about. So that's very important. And there are some viral co-activating molecules. They do not bind to the DNA, but also can modulate transcription, and we are going to see that. And also there are some co-activators that structural activity modulates the nucleosomal template, and this is the take-home message from this slide, that the nucleosome structures basically can condense the DNA or loosen up the DNA. This is what we call as the euchromatin and the heterochromatin. So for the RNA polymerase to access the sites of transcription, the DNA needed to be loosened up. Remember that. The DNA need to be loose so that you can access that. I think that's pretty, normally it will come to that, come to your understanding, like a common sense. So to do that, what it is needed, the histones, these proteins which are making these nucleosomes, these proteins needed to be either acetylated or methylated. Remember that. They needed to, and these are done by histone acetylases or histone methyltransferases. So they needed to be acetylated or methylated. On the other hand, so acetylation and the methylation will loosen up the DNA. On the other hand, what happens, when you condense the DNA by tightly wrapping them around, the histone deacetyltransferase takes these acetyl groups out And the deacetylation will condense the nucleosome and not condense the nucleosome, will condense the DNA on top of that. And at that time, if the DNA is condensed, the RNA polymerase will not be able to access that. So that's basically is the suppression of the transcription. Loose DNA, acetylated, methylated, ready for transcription. Condensed DNA, deacetylated, demethylated, not ready for transcription or suppression for transcription. That's your take-home message from this slide. The next part that what we are looking at, that this is a sequence-specific transcriptional activator protein. Remember that what we talked about, these histone modification, this is all throughout that is happening. Whereas here, these are the specific protein properties that binds to those specific sequences. So the DNA binding sequences have some specific features. They have a structure called a zinc finger or a helix-turn helix loop so that they can just home onto the double helix. And also, they are basic in nature because your DNA, as we know, nucleic acid, phosphate, they are ionized and they are acidic, so basic amino acids will attract them. So these are the DNA binding domain properties of those proteins. Then the important part is the dimer formation. Most of these DNA binding proteins works as a dimer, and it kind of like it sits on the DNA like a fork. So you have one side, one monomer, another monomer comes on the other side and just wraps around the DNA. And the zipping function is done by what is called a leucine zipper motif. This leucine zipper motif, of course, you have these leucine residues in specific sequences that helps in dimerization of this transcription factor. And then you have an activation domain, and this activation domain may have acidic amino acid residues, sometimes glutamine rich, proline rich, isoleucine rich. These are some of the factors. So what do we mean by the activation domain? This is the binding site of other proteins or stimuli related proteins which can activate the function of this protein, basically. So basically what these proteins are basically doing, they are making the RNA polymerase to start. Without their function, the RNA polymerase cannot start. So just think about that. You have a full gas tank of gas in your car. You are sitting in there pressing the gas, but unless you just turn on the key, you just cannot. So you turn on the key, still you press the gas, it doesn't start. So when you turn on the key means that, okay, all right. All the transcription factors are sitting on that RNA polymerase in there. You turn on the key to one position, it's not starting yet. Then you crank it, at that time means that the activation domain is activated and the RNA polymerase can start. You can think about that analogy, basically. So what are the different strategies? So you can see that the host only, that the retroviruses and the simple genome like a colimovirus or circovirus, their transcriptional program depends upon the host. On the other hand, for these cases, you need the host transcription factors or at least one viral protein control their transcription. Say bacteriophages, the late genes, and also for hepatinoviridis, parvo, papilloma, polyoma, retroviruses with complex genome, you need at least one. And you can see that for the large viruses, fairly large viruses, adenoviruses, herpes viruses, greater than one protein is needed for the transcription. Whereas, again, for the pox viruses, giant viruses, all of the transcriptional factors are viral and they have their own DNA-dependent RNA polymerases. And what is the advantage of that? They can just do the transcription, the whole thing in the cytoplasm. It doesn't even need to go into the nucleus. So that's the advantage of these. So what are the different strategies that what we are looking at? That this is the process by which the genes of the virus are activated. So first, what we are looking at, that right here, you have a gene X initiator sequence. It makes the gene from the gene X is transcribed into viral mRNA, which is translated into the protein X. These protein X can bind to its own initiator sequence, and this is what we call as a positive autoregulatory loop. And this is basically nothing else than a positive feedback loop. And it's just going to make a lot of transcript, and that's what it is. Remember that instead of a positive autoregulatory loop, you can have a negative autoregulatory loop. That means, and that could be a question, a critical thinking question for your exam, that suppose the protein X is binding, suppressing the function of gene X, or it's not making the mRNA, then this positive autoregulatory loop will be called a negative autoregulatory loop. Both happens. On the other hand, this is called a cascade regulation, where the first protein X moved and the protein once made binds to another initiator or regulatory sequence of another gene and controls the transcription of that. This is called a cascade, and these transcripts can make another protein and control the Z and so on. That can happen also as well, and that's what we are going to see. So here we are looking at the polyomavirus, and what we are seeing right here, the genome is first making the transcript mRNA, and it goes into the cytoplasm and it makes two proteins, an LT, large T antigen, and an ST, small T antigen. This LT is transported back into the nucleus, acts as a transcriptional enhancer or regulator, makes the second round of mRNAs, which makes this viral structural protein. So this is the simplest control, which are the structural proteins. So you can see that this is a temporal control. Initially, it makes the mRNA to make the transcription factor. This transcription factor now makes the viral the proteins after some time, because you don't need the structural protein at the very beginning. So this transcriptional control does that. It temporarily separates when the virus needs what. They do not make all of them at the same time, because that will be a wastage of energy or efficiency. So here what we are looking at, that this is an SV40 transcriptional regulator. So think about that from the left to right, that black line is the timeline when the genes are expressed. So at this point, at the very left, this is the start when the genome is delivered, and the L protein basically is made after that in the early phase. Once the LT or the large T antigen is made, it enhances DNA synthesis, and it does what is called an anti-repression. That initially these areas, which are the late genes, they are not expressed. That means that they are repressed by some repressors. These are the proteins which bound to this L region, and this doesn't allow them to transcribe. So the LT allows them to escape that repression to enhance them. On the other hand, initially the LT is made, and it also blocks the initial early transcription. So again, remember that carefully. In the early phase, LT is made. Once LT is made, that is the only function of the early area. You do not need an LT anymore. So it stops the transcription from this part early. Now the LT enhances the DNA synthesis as well as the anti-repression. This L region, or the late region, makes all the proteins of this VP1, 2, 3, 4, which are the structural proteins, and also make the genome to be packaged into the virus. So that's the viral DNA synthesis, which is happening right here, and this. So LT is doing both of this. So there is another mechanism that happens with the SV40 late promoter. There is a cellular protein called an initiator binding protein. It's a thyroid steroid family of proteins, cellular function completely. So basically what happens that question is that you may ask that what is this anti-repression? How this is not expressing, how this is happening? This is what the answer is. So this sequence, the whole thing, is basically the regions of the SV40, which is the late region. These are the late genes, which codes for the structural proteins. When the genome is exposed into the nucleus, this IBP, or this initiator binding proteins, bind to their control region, and this is the repressor molecule. It just binds to them. And see what is happening, that after the initial expression of this LT, it increases the viral DNA synthesis. See that it increases the viral DNA synthesis. So what happens that the viral DNA increase. So here, this is the initial amount of DNA that is delivered into the cell. Everywhere, the repressor is binding, and it is not allowing the late gene to express. As DNA synthesis increases, they run out of the LT, and they cannot be covering those anymore. And this is what we call as an escape from the repression. So that means that this is basically anti-repression or escape from the repression to allow the late gene expression. See that this is how it is happening. The amount of IBP in a cell is constant. That's why you are seeing a single line. So remember this graph very carefully, because we will have some questions from this area. So you have a straight line but right here, which is the amount of IBPs. On the other hand, what you are seeing that initially, you have less amount of viral genomes. This is just the genome that came in. There is not much genome in there. Once the LT, or the large T antigen, is expressed, it is enhancing the expression of the DNA, or enhancing the synthesis of the DNA, as we have seen, more DNA. As more DNA is produced, there is not much IBP available to cover their regulatory sites. And that's why the RNA is now being made, and your late transcripts for your viral structural proteins are made. And this is basically the transcriptional control for that. So we will stop here, but there is no need to rush, and then we will go from there in the next class. We have just two of them are late, but I'm just taking a little bit of more time, because this part is complicated. Yeah, so you'll take some time. So definitely do your quizzes and just read ahead a little bit. So that will help you guys a lot.